Once we have our microbes in focus using the high dry lens, we might be ready to move up to the oil immersion lens. We might not be using the oil immersion lens in the chapter 5 experiment. We might wait until chapter 8 when we do smears and stains before using the oil immersion lens, but we'll talk about some of the basic steps. So again, we want to make sure our specimen is in focus with a high dry lens. I'm then going to rotate the turret just to get the high dry lens out of place, and I'm going to use some high quality immersion oil, and I'm going to place a drop of the immersion oil right on top of my specimen. Now this specimen has a cover slip, so the immersion oil goes right on top of the cover slip, right where the light is passing through my specimen. Later, when we do bacterial smears and stains, we won't be using cover slips, and we will put the immersion oil right on top of the stained bacteria without a cover slip. Now again, watching from the side, we're going to rotate our oil immersion lens slowly and gently into place. We want to make sure it's not going to ram into the slide. And for the oil immersion lens to function properly, it has to be touching the oil. It has to be immersed in the oil itself. So we need to have the tip of the oil immersion lens just touching that immersion oil. The reason for this is that we're trying to reduce light refraction, the bending of light. Light, as it moves from glass into the air, will change speed, and as a consequence, it's going to bend or refract. Now you recall how tiny, but a little diameter the oil immersion lens has. So any light that's refracted or bent means that we're going to be losing photons of light that won't enter the oil immersion lens, and thus we're going to decrease resolution. What the immersion oil does is it replaces that airspace with oil, and the beauty of that is as the light moves from glass into the immersion oil, there will be a very tiny change in speed. And as a result, the light will not refract, it won't bend as much, it'll travel more in a straight path right up into the oil immersion lens. So we'll be harvesting more photons of light that will strike your retina, make more action potentials, more data point for the visual center of the brain. So we're going to increase resolution by using the immersion oil. So if one was to ask, why do we use our immersion oil? The immersion oil decreases refraction, and as a consequence, we're going to increase resolution. If you were trying to look at your microbe using the oil immersion lens without using immersion oil, it'll drive you nuts because all you'll see are these fuzzy, blurry images. As soon as you replace that air pocket with the immersion oil, then you'll have nice, beautiful resolution. You'll be able to see the clear outline of your cells.